So we're going to continue and talk about what S phase actually is. So in the last lecture, we talked about how we have this skin cell. Let's switch. That's supposed to be a circle. And inside the cell, this is the nucleus. Inside the nucleus, we have 46 chromosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. One, two, three, four, five, six, 7, 8, 9, 10, 11, 12, 13. The reason I always say 23 pairs of chromosomes is because you have one chromosome from mom and one chromosome from dad for each trait that you code for. We're going to go later on into what that actually means when we go on and talk about Mendelian genetics. But today, we're just going to talk about how these cells replicate their DNA. So currently, our cells have 46 chromosomes. These are our body cells. Oops. 46 chromosomes. You can think of them as 46 chromatids. We're not technically supposed to think about it like that, but it's much more intuitive, I found, if you think of your body cells starting with 46 chromatids. And when the cells actually replicate their DNA, let's see if I can draw this. Oops. Here we go. So when the cells replicate their DNA, actually, let's see if I can. See if I can figure out. Is there a There we go. So when the cells replicate their DNA, it's actually better that I draw it. So if we take a copy of this DNA here, let's say we take a chromosome. Let's say we zoom in. Let's say we take a copy of this we zoom out, zoom in, I'm sorry. So in this photo, we actually have, here's a chromosome. And each chromosome, we can know it's a chromosome because if we count the centromere, there's actually centromeres on each chromosome. So when I draw this out, assume that there's these central knobs called centromeres. And I'm not going to do it for each one, but you get the picture. So this is before replication. So before DNA replication, we have 46 chromatids. We can count the chromatids by counting each leaf-like projection. This is one leaf-like projection. The green over here is one leaf-like projection. The other leaf-like projection here is the pink. We can count the chromosomes by just counting the centromeres. So in this example, we have 46 chromosomes because if we counted each centromere, 1, 2, 3, 4, that's the black part, the black circle. There's 46 of them. So there's 46 chromosomes. If we count the leaf-like projections, we have 46 chromatids. So I know what you're thinking. You're thinking, so what? Is there a method to all this madness? And the actual answer is yes. Because when our DNA replicates, notice that we have when our DNA replicates, we have still the same number of centromeres. These are the same number of circles, black circles. So after DNA replication, we still have the same number of black circles. That's the centromeres. So after DNA replication, we still have 46 chromosomes because if we counted these centromeres, we'd still have 46 chromosomes. But notice that if we counted the leaf-like projections, now instead of one leaf-like projection over here on the left side, we have two leaf-like projections. So we've actually doubled the number of chromatids. So we have 92 chromatids. So our body cells always have 46 chromosomes, but they switch between the number of chromatids. And so now that we've copied our DNA, we can go on to S phase. We are in S phase. I'm sorry. We can go into G2 and then eventually divide into cell in mitosis.